the meeting settings, but I don't think it's too much of a problem. Hopefully everyone's just muted their mics and I'll just keep going on as normal. So with this one, guys, I did see a few people think that the options would be out of 4. There's 4 different poles here. But if homozygous recessive isn't compatible with life, we don't actually include it in our expected ratio. So actually the ratio would be out of 3. So you'd have a 1 to 2. You'd have 1 for homozygous dominant, and then the 2 would be heterozygous. So I hope that's clear. And then moving on then. So this is an example of a dihybrid cross. I've done an example to you here. And as I said, it's the inheritance of 2 separate genes. And we'd only do a dihybrid inheritance cross if the genes are unlinked to each other. So I mentioned earlier Gregor Mendel and his pea plants. And so this is a really nice example of what he actually found out. But of course, he didn't know what an allele was per se back then. He just noted down the appearance. So you've got round yellow, wrinkled yellow, round green, and wrinkled green. So these are the different combinations. And as I said earlier, you can have in a dihybrid cross, a heterozygous dihybrid cross, you'll have this expected ratio at the bottom, 9 to 3 to 3 to 1. And so that's just an example for you. And I want you guys to now attempt this exam question here. It is a dihybrid cross. So I'm going to give you a few minutes to have a go at this question, and then we'll talk through it. And you guys can put your answers in the chat of what you expect the parental genotypes to be and the gametes to be.
So red are females.
So I'll just give you one more minute to have a go at that question. And then if you don't mind putting it in the chat what you think the answer is. Okay, so let's work through this question together then. So first of all, it says at the top of the question, a mutation. So I have taken this as like a part B of a question, but the first part isn't relevant. But let's work through it. So a mutation in another gene at another locus in Drosophila gives rise to white-eyed flies. The red eye allele of this gene is R, which is dominant, and the lowercase r is the white recessive allele. So students crossed these red-eyed flies with a white-eyed fly, and they've got the table below showing what they've found out. And so what we're trying to do is we're trying to work out what this cross would be in a typical cross. So you can see, first of all, that they've mentioned big R, little r here, but then they've also mentioned X and Y. So instantly in your head, you should be thinking dihybrid cross because we've got 2 separate genes being mentioned here, okay? So the rule of which we'd go by is we'd go with putting the sex chromosomes first and then referring to any other kind of allele afterwards. So we would expect that the parental genotypes would be for parent A, XR, XR. And that's because we can see here that they said that males are heterogametic. So they've got different sex chromosomes, whereas females will be XX. So that's why we've got X little r, x, little r. And you can see that here in this table here, okay? Because again, the table isn't showing what we would expect. It's showing what's observed. So that's a big difference, first of all. So don't get confused by that in the question. The table there can throw some students off, if that makes sense. So we've got here that the student crossed a red-eyed fly with a white-eyed fly. I will go through the cross in the next slide, but I want you to just look at this table and like kind of not let it throw you off, because it can be quite confusing looking at this and then all this information. So first of all, like I said, we've got the males are heterogametic, so that should be ringing in your heads that that's going to be two different gametes, and there's already two different chromosomes. So that should be saying X and Y must be male. Therefore, it hasn't told us what females are, so we can assume that they must be XX, right? So that is the first part of the question, and that's just referring to one gene, okay? But then in the top of this question also refers to big R, little r, okay? So that's the other gene that we need to take into consideration. That's why we need to think of this as a dihybrid cross, okay? So that's why on the next slide then, I'll go through it with you. We've got XR, XR for the female, and we've got X dominant R, Y for the male. And as I said, the rule of thumb is like the way you would approach this typically is you do the sex chromosomes first, and then whatever the autosomal chromosome is after that, okay? So that's the way we'd approach the parental genotypes. You then split this up essentially into thinking what combinations of this genotype here will be passed down into the gametes. Remember, gametes are haploid, okay? So they'll code for half of the genetic material essentially. So in the female's case, because they've got two of the basically the same, all their gametes will be XR, whereas with the male, because they only express the big R and not the little r, they'll be bound together. If you split this up, essentially, it's like drawing a line in half down the middle, yeah? Think of it like that. That will split up into two separate gametes, but they'll be the same. So that's why in the mark scheme, they've only just put XR, because that's all the combinations you can get. Whereas with the parental gametes for the male, you'll inherit the Y chromosome by itself because it's not expressed with the alleles we were talking about before. So it's only on the X, okay? And then you've got X dominant with a R dominant here, okay? And then you think about how these will cross over when you mix gametes here with the gametes here, okay? And that's why you've got these genotypes here. So I hope that makes sense. What I'll do when I publish these slides, I'll draw the cross as well, because obviously the mark scheme can be a bit confusing, but I'll draw the cross for you as well. So hopefully that'll explain a bit more. But yeah, so that's what you should have got for that question. And then moving on, so that would always be the part A of a question in the exam for both OCR and AQA. They would only give you a dihybrid cross if they were going to follow it up with a chi-squared. And so that's what I've gone through here. Again, referring to Mendel and his P's in a dihybrid cross, I've gone through with you. So these are the numbers that were observed. So for example, let's say Mendel did his pea plant experiment on scale, and he observed that 315 of the plants were round yellow. So the peas were round yellow. 108 of them were round green. 101 were wrinkled yellow, and 32 were wrinkled green. That is giving you your observed number, okay? So with chi-squared, you're using, as I mentioned in the slide previously, you're looking at whether the observed number correlates statistically with the expected number. And the way we do that is by following these steps here. So first of all, we add up all of our observed numbers, to get 556. And then we divide by 16 because, as I said, what's the rule of thumb with a dihybrid heterozygous cross? You'll always get this ratio up here, 9 to 3 to 3 to 1, okay? So that's why we've got 16, because you add all these numbers up and that's 16. So that gives you one part of the expected ratio. So 35 would be the expected ratio for wrinkled green, which is what I've done here, okay? And then you would do this 35 and times it by 9 to reflect how many expected round yellows you get in this cross. You do it by round green, and because this is 3 and this is 3, you'd get the same number for both, so you get 104. So in an expected dihybrid cross, you'd expect to get 104 round green and 104 wrinkled yellow pea plants, which is what I've shown here. And then the next step you need to remember is that with chi-squared, the way you do it is by using this formula here at the bottom to calculate the critical value, okay? Which this sigma here means the sum of, and then you do observed minus expected squared divided by expected, okay? So this table clearly illustrates the observed here minus the expected, which is what we calculated up here. We've got our differences. We do the sum of our differences over the expected value. And I've broken that up to just kind of show you even more clearly where all these values come from. And you add them all together, you get 0.51, and that's your chi-squared value, okay? And then in the exam, they'll give you either a formula sheet or they'll give you a table as part of the question. And what you need to do is you need to compare your 0.51 or chi-squared value to the degrees of freedom and the number that they've calculated there. And you need to remember that as n minus 1, with n being the number of categories in this case. So we've got 4 categories. It would be 4 minus 1. That would give you 3 degrees of freedom. You then follow it along on the table that they've given you, on the statistical sheet that they've given you, and compare your 0.51 with the value that is on the chi-squared statistical sheet for you. And so then if your chi-squared value is less than the one that you have looked up on the statistical sheet, then it's acceptable and it matches the null hypothesis, meaning that there's no substantial difference between the observed value and the expected value. And that kind of makes sense if you can see, because we're quite close. The observed value here was 315 and you expect to get 313. So you can tell in this one, there's no massive statistical difference between the observed and the expected. And that's basically what chi-squares tells you. And if there is a big statistical difference, you know that there's something else coming into play here. You know that there's another factor coming into play with this cross. And so then you would reject your null hypothesis, and that would be your conclusion, essentially. And that's all you have to do for a chi-squared, really. So I hope that my walkthrough there has explained to you how you'd approach a chi-squared test question. And I want you guys to have a look at this one and do, using your previous dihybrid crosses that we calculated a few slides ago. So this basically here, if you can make a dihybrid cross from XR, XR, XRY, you can kind of get a feel for what you'd expect here. And I put this table here because this table here is the observed values, and we need to calculate the expected. Read the whole question carefully because this one's quite a complicated one. So I'm going to walk this through with you because it may not make sense initially, but hopefully with my explanation it will. So read this part here. Remember this table. This is the observed values that the student noted down. So if you can see that there's an observed value of 0 and 0 here, we're not going to be using that. And the examiners have actually set that up for us. They don't want us to go wrong and waste time. So they've only set it up for the red-eyed female and the white-eyed male. And it's also told us in the top of this question, which could be quite easily missed, the expected ratio of red-eyed females to white-eyed females is 1:1. Okay, so think about that. Think about how that influences our expected values here, okay? And then these parts should come quite easily once you've worked out these two. Okay, so I'll give you a few minutes to do that. And as I said, the conclusion is, once we've calculated our chi-squared value here, we've only got 2 categories here. So we're going to do 2 minus 1, which means our degrees of freedom is 1. And then we're going to scout along. We're going to go to, it depends on which statistical value they want us to use, the probability, but it's usually 0.05, okay? So we're going to use this value here. I've given you a hint there already. And you just need to compare this value here, 3.84, with your chi-squared value, and that'll give you your conclusion, whether there is a statistical difference between the observed and the expected values, or there isn't a statistical difference, okay? So I'll give you a few minutes to have a go at that, guys. And then I'll walk through it. Yeah, so it is different in chi-squared. So I get what you're saying here with the p being less than 0.05, you would reject the null hypothesis. Whereas in this one, if it's less than the probability value here, then you wouldn't reject the null hypothesis. In chi-squared, it's different. It's the opposite to hypothesis testing. And then with the monohybrid, yeah, so as V put here, it's the opposite for there. And the genotypes, so the genotypes that we needed were the ones we calculated before. But to be fair to this question, you don't actually really need the stuff we did before for this, because it actually gives you everything here, because it doesn't want to help, it doesn't want you to carry any errors forward. It wants you to be able to access all the questions isolated of each other. So you can actually use everything here, this 1-to-1 ratio here, the number of offspring here, and hopefully that should give you some help there. So the dihybrid cross is not actually linked to this question, but it's trying to help you understand the cross here. Basically, I wanted you guys to get a better understanding of why there aren't red-eyed males or white-eyed females. That's hopefully why it'd help you with the dihybrid cross. So I hope that explains a bit more why I put here using your dihybrid crosses. Hopefully that makes a bit more sense. So I just wanted you guys to understand the cross here and how it's resulted in these offspring here. Yeah, okay, so let's go through this question then. So our observed value for the red-eyed female would be 27, the white-eyed female would be 23. Now our expected value, because it's a 1-to-1 ratio, whatever number you'd put in for your expected, it would be the same. So in the case of this question, let's just add the ratios together, get 2. So then you'd have two expected red-eyed females and 2 white-eyed males. You'd then do the observed minus the expected and square that. So the examiners here, they've given us a table and they've gone through and they filled it out themselves. So this mark scheme is quite confusing. I will admit that. So the 2 comes from the ratio being added together, the expected. So you would have 25, but even if you use your values, which is what you'd expect, like I said, this mark seems very confusing because I think it's trying to approach it as a simpler way. But your values, no matter if you put 27 and you minus the 2 and you've got 25 here, and then you minus 2 and you've got 21 here, all these different values following on would still be different, and you should still be getting the same difference, and then they would still come to the same conclusion. So they've just used these values here for simplicity, but this is how you go about it. I'm not sure why they've done this, but just go about it the way I ran through it in this example previously, using expected values. I'm not sure why they've done it like that, but let's just remember that even with these values, you still get a chi-squared value, which would mean that you would compare it in the table and you wouldn't reject the null hypothesis. You would say there's no statistical difference, even if you use different values, okay? Because you still remember this 1-to-1 ratio here, so it'd still work out, okay? So this question is from OCR. And in case you guys are wondering, so it's an OCR question, this one. So I hope that's made sense. If not, I'll answer any questions you have on chi-squared and the exam questions we've gone through at the end. Just for the sake of time, let's move on to populations, OCR 6.1.2 and AQA 3.7. And this is what we're mainly going to focus on. Point 1 here. We're mainly going to focus on Hardy-Weinberg, only because that's the main point that gets tested on. It's quite easy to test it. It's testing your maths, it's testing your understanding. And you need to remember that with Hardy-Weinberg, it assumes no mutation, migration, selection, genetic drift, or non-random mating. And so I've kind of explained to you here what genetic drift is. So genetic drift is more pronounced in small populations. And that's because there's a restricted gene pool. And gene pool refers to the frequencies, well, the availabilities, I guess, of different alleles. So think of a population of 100 people. If there's a small population there, they're all going to hopefully evolutionary mate with each other and have kids. The genetic pool will be quite restricted. And because of that, the genetic drift, which is basically the frequency of certain alleles, will become more pronounced. So because there's such a restricted gene pool, you'll get a lot more pronounced alleles and expressions of that in the phenotype. Gene flow, that's just introducing new alleles to population. So in the case of that 100 people, say we had about 50 migrate from a different population, that's gene flow. They would expand the gene pool. Genetic drift wouldn't be so pronounced then. And yeah, there'd be a different frequency of different alleles present there. Selection pressures, that's really important to note. And this is for evolution as well. You guys can read that there. And basically, these all link into natural selection. So selection pressures are basically, let's use this case study here. So melanism. So in peppered moths, they were initially white, but then because of the Industrial Revolution, a lot of soot was being produced. And what's it called? They would stand out quite easily to predators. So because of these environmental changes, and then of course the predation as well being a selective pressure, what they did is that over time they evolved to change their color from being white to black peppered moths. And so that's an example of how selection pressures through natural selection can lead to organisms with more stronger and more survivable and advantageous alleles to future generations. So sickle cell anemia is another great example. Sickle cell anemia is an example of how malaria is a disease that can bind to red blood cells, and then of course spread and cause havoc in the system, in the human system, the human body. But with selective pressures, obviously people dying because of malaria, an advantageous allele developed in which you'd have sickle cell red blood cells, and they wouldn't be as great carriers for malaria. So those people would actually be resistant. And so you can get people who have normal hemoglobin, so HbA, and HbS, which is sickle cell hemoglobin. And these people, about 50% of their red blood cells are sickle cell, where the other 50% is normal shaped, and they have some resistance to malaria. Whereas people who have HbS, hemoglobin sickle, they will have a more pronounced resistance to malaria. But as a result of that selective pressure, yes, it was advantageous to be resistant to malaria, but it's now resulted in sickle cell anemia, which is a very prevalent disease, especially among those of African descent, because malaria primarily affects the continent of Africa. So Hardy-Weinberg, as I said, is the main thing that examiners test you on about this topic. So you'd use two formulas, P plus Q equals 1, then P squared plus PQ plus QP plus Q squared equals 1. So this is the frequency of the dominant allele, with being P, and Q is the frequency of the recessive allele. Now, as you know from the few slides ago that we did on inheritance, you can get people who are homozygous dominant, which would be represented by the P, homozygous recessive, and then you've got heterozygous, and that can be, let's say, P is big R, Q is little r. That could be big R, little r, or that could be little r, big R, and the combinations would just add together to get 2PQ. So that's why you use both P plus Q equals 1, and you extrapolate from that what the frequency of a heterozygous person would be. So I've gone through an example here. A particular characteristic is a result of a recessive allele. And we know that that particular characteristic is exhibited by one person in 25,000. 1 in 25,000 is this number here. And because it's representing Q squared, which is the combination of all recessive, I've just plugged that into my equation, and we know that equals 1. You do a little bit of rearranging, and you also use this formula up here. You can work out, as you can see from all my workings here, the different values. So I hope that this little walkthrough here makes sense. So you've got P squared being the homozygous dominant frequency, Q squared being the homozygous recessive, and 2PQ being the heterozygous. And I've gone through that example there. Okay. Is everyone okay with that example? Understood? Lovely to see. I'm glad. And then we can move on now to using this exam question. This one's a lot easier, guys. So I hope you guys are all able to answer this one quite nicely using the example I walked through with you earlier. I'll give you 2 minutes until 6:48. See if you can have a go. But just to point out, dominant allele B results in brown fur. So make sure you remember that when you're referring to this 45 here, you're referring to dominant. And make sure you plug it into the right equation as well. That's my other top tip for you. So if everyone's had a go at that question and starting to get an answer, Do you want to maybe put it in the chat so I can see? Yeah, very happy to see 0.5. Lovely stuff. Nice, guys. Yeah, so 0.5 is the correct answer. And does anyone actually feel confident enough to unmute themselves and maybe explain why it's 0.5? No one feels confident enough? No worries at all. I'll explain to you now. So first of all, we know that there's 60 rabbits in total. So that's 60 rabbits is essentially representing our one here. We then go and we realize that 45 of those rabbits are representing the dominant allele, so BB. So 45 over 60 will give us 0.75. And that's basically your proportion of dominant. So you've got 0.75 being dominant and 0.25 of those rabbits being recessive, so being albino. And then from there, you can extrapolate by using this formula here, by plugging in your values. So you'd use your, bear with me, let me just get my calculator. Bear with me. Do you want to get it on a whiteboard, is it? Okay, I'll put it on a whiteboard if I can try and get it. If you want, actually, what I'll do, I'll run through all these questions at the end. I'll come back to it just to make sure we've covered through all the content. What I'll do, I'll go through this cross here on a whiteboard, this Hardy-Weinberg, sorry, on a whiteboard at the end, right at the end, all right, guys? Just so we make sure we get through all the content. So, yeah, so there you go. So Arish has put it in. 0.75 is the p squared. You'd square root it, and then you would be able to work out then what your p was and your q. Okay, but I'll do that on a Jamboard for you after this. Let's just go through. So I've included it all here, but I'll write it out with you all, guys, if that makes more sense. Okay. So this is another one, but again, if you've got more time later on, I'm conscious of time. Let's just get through all the content and then we'll approach all these exam questions at the end, all right? So moving on, following on from populations. So we talked about how Hardy-Weinberg can tell us about the frequency of an allele within a population. And it's important to know how, you can get, when I was referring to that group of 100 people, how you can get genetic drift and how those 100 people, let's say they're stranded on an island, yeah? They are actually, if they continue stranded on that island and no one, lands on a boat and brings more genes with them and then they mate with the population, they're not bringing any new genes in. So no gene flow is happening. And because they are, let's say, what's separating the island is a really dangerous and volatile sea. So they can't cross it. They can't get in. Yeah. They can't get into the island. So that would be allopatric, or let's say that's geographic isolation, and that would eventually lead to allopatric speciation, because basically that group of 100 people, they're mating with each other, and eventually they will become so different to let's say a group of humans with 100 people. They become so different to, like, let's say people living in the UK, because they've been separated for so long and they've been geographically isolated. They would actually become a new species. Whereas with sympatric speciation, that can be brought about by mutation, and that can affect different people. Let's say that can result in a change in the sexual reproductive anatomy of people. So that could mean that a species may become infertile and they may not be able to mate with each other. Or let's say they have a selective advantage, which means things won't fit, so they won't be able to mate properly. Loads of different factors can affect reproductive isolation, and that can lead to sympatric speciation. So it's important to note the differences in speciation here and how they can be caused by either geographic isolation or reproductive isolation. And that's basically the big chunk on populations and inheritance. To summarize, they'll mainly ask you questions on Hardy-Weinberg and dihybrid crosses and chi-square tests. Those are the three takeaway points. If you didn't take away anything from what I've gone through, take away that those are the three most important things that they like to test you on. And now I'm going to move into mutations, which is basically, that is basically directly linked to inheritance. Mutations can be inherited and change things and lead to speciation, actually. So it's good to actually look at what kind of mutations occur and what they are. So mutations are changes in the genetic material that can occur at the gene or chromosomal level. I've listed a lot of different types of gene mutations. That's for you guys to read in your own time, I guess. Chromosomal mutations, that can involve structural changes or numerical changes. So people are aware of Down syndrome, that's trisomy 21. That's resulting in the inheritance of 3 chromosome 21s. And that leads to, the symptoms of Down syndrome. Spontaneous mutations, they can arise during DNA replication. And these are mainly linked to quite bad things. So, cancer, skin cancer, melanoma, that's usually linked to UV radiation and exposure to that. Viruses can increase mutation rates. So HPV, human papillovirus, that can be very bad. It can cause cervical cancer in young girls, which is why most people in here are probably vaccinated against it in school. But like I said, not all mutations are bad, and mutations can introduce genetic variation essential for evolution. If you look at us compared to humans 1,000 years ago, we've mutated quite differently to them, and we're still around. So obviously those mutations must have been beneficial. As I said, natural selection, Charles Darwin's theory of, the strongest and most advantageous alleles get passed on, and that's what leads to the survivals of a species. Mutations are, of course, impacting the genome, the human genome, and the genome obviously is DNA, which is deoxyribonucleic acid. You guys know the double helix and what DNA looks like. And you guys need to be aware of how DNA can be regulated and how that influences gene expression. So it's important for you guys to note that promoters increase transcription rates while silencers decrease them. So that's something for both OCR and AQA. And that influences where you guys should all be aware of RNA polymerase and how that will bind to and what transcription factors will cause RNA polymerase to bind to and ultimately result in mRNA being made, which is the transcript, and that's the process of transcription. And then obviously that transcript, that mRNA will leave the nucleus, it'll get to the ribosome and it'll be translated. And that will result in a functional protein being made in normal circumstances. Now, transcription and translation can be affected on the DNA level, and that's mainly through, as I said, promoters and silencers, but also it can be epigenetic mechanisms such as DNA methylation, histone modification. I've got another box here, how methylation typically silences genes by blocking promoter regions, preventing all that transcription machinery from binding onto the DNA, resulting in mRNA being made. And then histone modifications that can activate or repress gene transcription, depending on whether DNA is tightly coiled around histones or it's quite relaxed around it. And that influences how the transcription machinery can bind to it and result in mRNA being made. Let's see if we can go through that at the end. But I just wanted to go through this slide with you here. So it explains very nicely what I just went through with you. So we've got our double helix of DNA here and how DNA is essentially coiled around histones. And as you can see, Here, it's coiled quite tightly around the histone, which means that DNA actually isn't accessible. Whereas in the histones here, it's not so tightly coiled, which means that the RNA polymerase can access that DNA and make mRNA. And epigenetics can be a result of many environmental factors. It can be diet, it can be aging, it can be exposure to UV, all sorts of things. But just remember that it's environmental. And here's a summary of DNA methylation and how a methyl group combine to DNA and impact transcription of that DNA. So this is for OCR only, and I'm going to quickly run through it. The transcriptional control of lac operon is an OCR example of how gene regulation can occur. That's for you guys to read in your own time, but I just want you to be aware that it's actually quite common for them to ask you questions on lac operon, so you do need to be aware of it when you refer to body plan and cellular control. If they ask you a question on it, they like to use it because it's quite a nice example in E. coli about how different parts of different substrates like lactose and glucose can control the transcription of beta-galactosidase. Transcription factors, we've mentioned those already. So transcription factors are basically what tell RNA polymerase to bind to it or not to. And that's basically with the promoter basically initiating transcription when it's present, or if it's not present, it means that RNA polymerase can't bind, it can't transcribe that DNA, and it can't make a functional protein later on. Post-transcriptional control. So you guys should be aware that mRNA control contains introns and exons. Introns are referred to as non-coding DNA in that mRNA strand, whereas exons, they are coding DNA in that mRNA strand. It's usually referred to pre-mRNA, actually. And then that mRNA, that pre-mRNA becomes mRNA by undergoing splicing, which basically means it cuts out all the introns and only leaves the exons, so that when it eventually goes to the ribosome, it can be translated and turned into a functional protein without anything else being there and disrupting that protein chain, a peptide chain, sorry. Post-translational control, again, this is OCR only, and that's the specific example they want you guys to know about. But post-translational control, that doesn't actually necessarily involve just adrenaline and adenyl cyclase. It can involve, just adding a glucose molecule to a protein or a sugar, and you can get glycoproteins. You can get all sorts of different kinds of proteins. They can be cleaved at the Golgi body, which I'm sure you guys are familiar with, and they can be edited there, packaged and modified. And that's an example of post-translational modifications and control. Again, OCR only, Hox genes. These are really important signaling and transcription factors that are essential in basically setting up the body plan. If you guys go on to do medicine, you will learn a lot more about Hox genes, but that's just something for you to be aware of, how they influence the body plan and lead to normal limb development mainly. Apoptosis, programmed cell death. This is different to necrosis, guys. Make sure you make that abundantly clear. Apoptosis is programmed cell death, whereas necrosis is cell death and resulted from trauma or disease or something which is outside of the programmed nature. Apoptosis is completely normal. We've got it going on all the time. We just don't notice because we've got mitosis going on as well in the background. So actually, you will have, during early development of an embryo, you'll have webbed fingers and webbed toes, but controlled and targeted apoptosis leads to that webbing breaking down. And that's why we've got our separate fingers and separate toes. Moving on now to the last part of this session, genetic engineering and biotechnology. This is probably the most important topic because examiners love to test people on this, because one, examiners are usually people who've done research in the past and they've used these techniques, and they like to talk about and set questions on things that they have done or that they know a lot about. So it's really important that you guys have a good grasp of genetic engineering and biotechnology. Here I've used the classic example of genetic engineering using recombinant technology. So recombinant technology is essentially outlined here. Hope you guys can see that properly. I know the colors aren't the best, but basically you would use a DNA probe. Say, for example, insulin actually is a great example of where we use recombinant gene technology actually. So say, for example, I'm a drug company and I want to make insulin en masse, okay? I want to make lots of it. So the way they do that actually is first of all, they know the code. They know where the insulin gene is located, which chromosome is on, and its code. So we'll get a DNA probe, which is complementary to that. The DNA probe will bind to the insulin sequence, which codes for insulin, that gene specifically. And then basically using fluorescent technology or some kind of imaging technology, you'll be able to see that. They usually have a dye attached to the DNA probe, so they know where the insulin gene is. They'll then use restriction enzymes to cut this DNA at specific sequences, creating sticky ends. And you'd use those same restriction enzymes in a plasmid, okay? So here you can see a plasmid with the host gene. It's being cut out with restriction enzymes. These same restriction enzymes are then used in another plasmid, and they are complementary, and they basically cut out a part for this gene here to move into, because the sticky ends will be complementary and will be able to bind to these bits here because the same restriction enzymes are used. And then you'll use ligase to essentially fuse these sticky ends together in the plasmid, and you'll get a recombinant plasmid here. So this recombinant plasmid, let's say it's got the insulin gene. Then what happens is you will put this insulin gene into a bacterial cell, and you'll let that replicate, and you will essentially give everything that bacterial cell needs to make insulin, and then you can harvest that insulin and you can get lots of it. So that's how pharmaceutical companies do it. But the thing you need to know about this, though, is that with these bacteria, if a plasmid contains an antibiotic resistance gene, say, for example, you've put insulin in it, but you haven't realized that it also contains an antibacterial, antibiotic resistant gene, obviously, that's not good. That's not what we want. They'll be resistant to antibiotics and they'll grow in the media, and they will, when you try to kill them off, they won't die with the antibiotic. So that's always something to be very aware of with using this technology, is that you can get antibiotic resistant plasmids. Moving on to PCR, polymerase chain reaction. This is really important, especially when you've got a tiny sample of DNA and you need to amplify it, essentially, get more of it expressed so you can figure out what this DNA is coding for. It's quite commonly used in the lab. It's used as well in crime scenes when you've got a little bit of blood. A lot of the DNAs, they're damaged, or it's not all in one like compact vessel, let's say. So you'd use PCR to essentially amplify the DNA you've got. You get more of it, and this diagram quite nicely shows you how it goes on. So again, using a DNA probe, you'd look for the target DNA sequence, it bind to it, and then you'd be able to know that that's the sequence you're looking for, okay? So you'd get markers and you put them here and here to kind of block it out. Then you denature it, which basically means breaking the hydrogen bonds, and that needs to be done at 95 degrees Celsius to break those bonds. The mixture is then cooled to about 55 degrees, and you put primers in, because does anyone know, if you want to put in the chat now, actually, what kind of replication is DNA replication? Is it dispersive, conservative, semi-conservative? Great, semi-conservative, right? So you use the initial strand, and then you get primers, or in the case of a nucleus, you'll have free-floating nucleotides, and that will complement the conservative strand. So you'll have one conservative or one parent strand, then you'll have one new one, which is why it's semi-conservative. And you'll put primers on, and that will essentially, as you can see here, give you basically the start point for free nucleotides to then bind to, because basically in semi-conservative replication, you guys won't know this at A level, but when you come to degree level, you'll need to know that primers are needed in DNA replication for nucleotides to see where the starting point is essentially, and then go from there. And then you'll have DNA synthesis occur about 72 degrees, because that's the optimum temperature for DNA polymerase to do its work by basically bringing these free nucleotides and catalyzing the formation of hydrogen bonds between the conservative strand and the new one. And this cycle is repeated countless times until you've got sufficient DNA to create a DNA profile, and that can be used to, say, for example, identify a criminal that you're looking for in the crime scene. Gel electrophoresis is the final experimental technique you guys need to be super aware of. And basically this involves a kind of jelly-like substance. Agar is usually quite neutral. Well, it is neutral, sorry. And you'll have little wells in which you can put DNA samples in. And basically you'll have a cathode, the positive, and the anode, negative. And basically, because DNA is negatively charged, the DNA molecules will form discrete band here, and they will spread out and they'll spread out and they'll form nice discrete bands, because the negative phosphate groups in DNA will move towards the positive electrode. And so basically the smaller the bands, the further they'll move and the faster. And then the larger bands, because they're so clunky, they won't move through the pores so easily. But they'll still band out, as you can see, but the smaller ones will form more discrete bandings. And so those three kind of technologies, those genetic technologies I was telling you about, are really, really important. So to summarize, recombinant gene technology, that's quite commonly used pharmaceutically. It's used to, isolate and use a desired gene, so insulin, for example. It's also used actually to make golden rice, which is something which is quite cool to read up on. Basically genetically modify rice so that it contains all the vitamins and nutrients you need in places where A, people aren't getting enough of it, and B, rice is easy to grow there. And it's also a part of the diet. So golden rice is a great example of where this technology here is used, as well as insulin. PCR, you guys do need to know that it's good for you guys to remember the numbers, 95 degrees, 55 degrees, and the 72. I've generalized them here, but this diagram is really good at giving you the specific numbers. And that's the same for AQA and OCR, guys, okay? And then gel electrophoresis, this is used to kind of separate the DNA, give you discrete bands, and basically, yeah, it's very useful for separating DNA molecules from each other. So we do have time, actually. I can see, actually, we have run out of time, but I would like us to try and go through this question here at least, only because it's a great question to talk about gel electrophoresis. So if you guys want to have a quick go at it, it'll only take a minute up of your time. Let me know what answer you get. Yeah, I was going to say southern blotting, that's an OCR, not an AQA one. You just need to know the three that I've gone through. I'm just looking through your questions. DNA ligase wouldn't have been used for PCR because we're looking to make two separate strands. And so DNA polymerase is the strand that you would technically use ligase actually after DNA polymerase has done its work. But yeah, actually you guys can refer to DNA polymerase as doing that work for you. I think in one of your exam boards, I'm not sure if AQA or OCR want you to specify DNA polymerase and ligase, or just know that DNA polymerase does that. Okazaki fragments, I don't think you guys need to know that for A-level. That's something you don't need to know for A-level, guys. That's just some extra reading, I'm sure of it. Taq polymerase, yeah, that's a great one to see. Seen it in mark schemes. Wow. Okay. Well, if that is the case, then I can quickly explain what Okazaki fragments are for you. That is something that I can go through quickly with you. And I'll use this. So first of all, let's go through this question and then I'll explain what Okazaki fragments are. So does anyone have an answer for this question? So it says lane 1 of figure 2 enabled the size of the different viral fragments to be determined. Suggest and explain how. Yeah, that's right. But does that relate to the question? Taq polymerase is the DNA polymerase, but it's specifically referred to Taq polymerase because it works at the 72 degrees that I mentioned. So you wouldn't use human DNA polymerase, you'd use Taq polymerase, which is a polymerase in bacteria. Lane 1 of known sizes. Yes, Naraman, this is great. So lane 1, guys, this is the control, basically. So these are your volunteers, and this is your control. So you've worked out the size of the DNA fragments, and you basically use lane 1 and compare it to lanes 2, 3, 4, and 5. So exactly, lane 1 into control. Lovely. So that's exactly what you'd say. So you get 1 mark for saying lane 1 is the control, and then you'd compare it against these, and that would be your second mark, okay? So let me quickly go back and explain what Okazaki fragments are to you guys. I'm sorry I wasn't aware that OCR does mention this, but it's quite important for you guys to know if you do take this further past A level. So basically, DNA synthesis is in a specific order. So you can see here we've got 3 to 5 3 to 5 prime of DNA with the 5 prime end and the 3 prime end, yeah? So every DNA molecule is anti-parallel to each other, and the way that is that you've got 3 prime and 5 prime complementary to each other here, and then on the other end, you've got 5 prime and 3 prime complementary to here. So the problem is, though, with DNA synthesis is that DNA synthesis occurs in the 5 prime to 3 prime direction. But you can see here, we've got a primer here. So this is great. So if we want it to be 5 prime to 3 prime, we're going to go all the way that way, and that's quite nice, yeah? But what's it called? We can't start off with this 3 prime here. We need to go 5 prime to 3 prime. So we're actually going to go the other way. So we're not actually going to synthesize DNA that can complement each other, right? So that's where Okazaki fragments come in. They basically fill in the gaps that have been left behind. So let me say that again. DNA synthesis is 5 prime to 3 prime, yeah? So you go in this direction. So basically imagine lots of nucleotides joining up, and they go all the way down and they complement this conservative strand here. But on this one, like I said, it's 5 prime to 3 prime, but we've got a 5 prime end here. So it actually go this way. It wouldn't go the other way, which is what we want it to do, right? Because we want a new strand to be formed here along this conservative strand. So what you'd have is you'd have Okazaki fragments fill in the gaps that are left behind, essentially. So I think that's all you should need to know for A level. Please let me know in chat if you need to know more, okay? But I hope I've explained that well for you. And I will add to my slides a slide on what Okazaki fragments are for you guys to look at a bit later on, okay? That happens in DNA synthesis. So just remember that for DNA synthesis. You can remember that in PCR as well. Yeah. Because PCR does involve DNA synthesis, so it's great to remember that. Yes, I did say I'd go through the Hardy-Weinberg. No worries. So you guys can do this exam question in your own time. And I will, like I said, guys, for all your questions in chat, I'll update my slides so you guys can see what I've found the mark scheme and the questions, okay? So I'll include the mark scheme for the electrophoresis question. But as I said, you'd get 1 mark for lane 1. You literally just have to say that's the control, and you know the size of those fragments. And then the second mark would be to compare it to all of this. So let me quickly go through this Hardy-Weinberg here, yeah? So let me see if I can get up a whiteboard on Zoom. If they let me. Oh, I can annotate, never mind. Great, okay. So first of all, we've got 45 is the dominant. Sorry for the handwriting, guys. I'm doing this on my Mac. So I'm just going to put 45 as the dominant, yeah? And then we know that we've got 60 rabbits in total in the pet shop. We've got 60 total here. So then we'd work out 45 over 60 as to be 0.75, right? 3/4, basically. Sorry, guys. This should all be year 13 content for you guys, not AS. This is A level. So if you do have any questions on that, just consult your spec, but it is A level only, all of this. This is year 13 content. Okay. And that's 0.75. You'd plug that in because we know it's the, what's it called? 45 of brown and 40 and brown is dominant. We'd plug in the remainder, which is 15 over 60, right? And so that would be 0.25. Yeah, and that's representative of the Q squared here. So we do the square root of 0.25. Sorry, guys, this is bad handwriting from me. And that would equal 0.5. And that is all they need you to do. Yeah, exactly. So 0.75 is the P squared, yeah. And you're looking for Q squared in this case, okay? So that's what it says on the mark scheme as well, guys, if you have a look. So yeah, you can see that 15 over 60 is the Q squared. And you would do 0.25 and you'd work backwards from that essentially. And then all you have to do is plug it in then. Once you've done 0.25 equals 0.5, you can then do Q and plug that in. And you get 1 minus 0.5, which is 0.5. So hopefully that explains it to you guys a bit better than the mark scheme could. Does that help, I hope? Yeah, okay, great. So I think we have run over a bit of time, guys. So I won't be able to take any more questions. But if you put all your questions in the chat, I'll see if I can amend my slides to match most of them. And yeah, I hope that helps. So these slides, I'll link them. I'll link the edited slides in the WhatsApp group chat. Okay, guys? I'll amend them and I'll include all the mark schemes, but they should all be in there. No worries at all, guys. I hope you got something from this session, and all the best with your A levels, all right? All right. I'm going to end the session now, guys. So like I said, I'll amend the slides to include stuff on Okazaki fragments, and I'll include the mark scheme as well for these. So best of luck with your A levels, guys. See you.